I've never taught my students anything. I just try to put them in the best conditions to learn. Albert Einstein. Diapath is based in Italy, near Milan, with a network all over the world. Diapath is a leading company offering a complete range of innovative instruments, reagents and consumables for anatomic pathology. Diapath Knowledge Academy offers educational webinars dedicated to lab scientists, anatomic pathology professionals, anatomopathologists and students. We are glad to introduce you our teacher, Dr. Antonella Savio. Dr. Antonella Savio has been working as a histopathologist for over 30 years in 5 different hospitals in Italy and UK. She will later introduce herself. Thank you very much for the precious cooperation. Before starting the webinar, just a few practical information. The webinar is about 30 minutes. Then we are going to have a question and answer session at the very end of the webinar. So you are kindly invited to have your questions at the very end, typing the questions in the chat for the final open discussion. Thank you. Now, let's start with the webinar, the pre-analytical quality in histopathology.
The topic of this webinar is the pursuit of quality during the pre-analytical phase. The pre-analytical factors include all procedures, practices and environmental factors to which a biospecimen is subjected before diagnostic assessments and tests are performed on it. High-quality diagnostics starts with high-quality tissue specimens, and guaranteeing a quality tissue specimen suitable for any further investigations is becoming more and more important in times of precision medicine.
What is precision medicine? Precision medicine is a term used to describe individualized treatment that involves the use of targeted diagnostics and therapeutics to the unique characteristics of a patient and of his/her disease based on genetic, biomolecular, or phenotypic characteristics. The concept of personalized or precision medicine represents a great innovation in medical care that has been historically based largely on a genetic approach to both diagnosing and treating disease. The principle is coupling diagnostics and therapeutics. In recent years, new advances in technology have converged with informatics to rapidly expanding this field. The molecular analysis of biospecimens is especially common in cancer care, where it often determines treatment choices and may also be used to monitor therapy in real time. In fact, the molecular assessment of a patient's tumor allows to underscore specific molecular aberrations and to implement a tailored therapeutic strategy based on this tumor's molecular findings. In precision medicine, the genetic change can be more important than the type of cancer that we are going to treat, because we can use the same drug targeting a specific change in different tumors. Similarly, different patients with cancers with identical morphology may obtain a good result with different therapies based on specifically detected genetic alterations. Cancers are inherently heterogeneous. whether or not there is a targeted or immune therapy available for any given subgroup of cancer, the molecular assessment is used to subclassify patients into more homogeneous groups according to the prognostic implications. This revolution started in the early 1990s, when the tyrosine kinase inhibitor imatinib, the Gleevec, was created to target the protein produced by the Philadelphia chromosome BCR-ABL translocation. When it was tested in chronic myeloid leukemia patients in a phase 1 study for safety, almost every single patient's cancer was improved. This rarely happens. Results from additional clinical trials were very successful, which led to Gleevec being approved as a chronic myeloid leukemia treatment. Thereafter, Gleevec was also demonstrated to be efficient to target c-KIT and PDGFR and has become the first-line treatment of choice for metastatic GISTs. This success story was one of the first instances when scientists understood what caused a particular type of cancer and made a specific drug to treat it. Another success story of personalized medicine was the development of a drug called trastuzumab, so-called Herceptin. In a proportion of all breast cancer cases, there are too many copies of a gene called HER2. This cancer, termed HER2 positive, are highly aggressive and often have a poor prognosis. Herceptin was developed to treat women with HER2 breast cancer, and it has made a big impact, such as improving survival for women with advanced HER2 positive breast cancers from 20 months to 5 years. There also can be too many copies of a HER2 gene in other cancers, including a proportion of stomach, esophageal, colon, and lung cancers. People with these cancers could also benefit from a therapy like Herceptin. Other examples of new therapeutics are medications that target altered genetic pathways in cancer, as vemurafenib for BRAF-mutated tumors, targeted immunotherapies for cancer, antibodies against immune checkpoint pathways like nivolumab and PD-1. This molecular approach is also producing a change in the way tumors are classified, as site and morphology are becoming less important now that we can use drugs targeting a specific molecular change in different tumors. For example, the drug vemurafenib, Zelboraf, is effective for patients with melanoma skin cancer that contains a specific mutation in the gene BRAF. However, this drug has also been shown to work for hairy cell leukemia and some forms of thyroid cancer also containing this mutation. In fact, nearly 2/3 of cancers bearing these mutations are tumors other than melanoma. This means that this drug could potentially be used for a wider variety of patients and many types of cancer. It is presumed that a molecularly tailored approach will increase effectiveness and decrease toxicity of treatment, decrease the costs of care overall, increase value in the health care delivery. It is clear that we are more and more required high-quality diagnostics and therefore high-quality tissue specimens. There are many variables that can intervene on the quality of the specimen and on the quality of the final tissue block on which we perform our assessments. Every step in the pre-analytical phase may artifactually alter the specimen and the final test result. The new challenge for pathology is to identify and standardize molecular-friendly techniques and technologies to be able to assure these requirements. But this goal does not involve only the procedures performed inside the histopathology laboratory after specimens reception. The pre-analytical phase also includes factors starting with specimen collection and transport. In addition to pathologists and lab scientists, the chain of custody involves as well surgeons, nurses, endoscopists, interventional radiologists, registrars, transport personnel, assistants, whose cooperation we need to count on to safeguard tissue specimens' quality. The overall molecular integrity of the tissue cannot be retroactively recovered once it is compromised. It must be safeguarded up front by control of the most problematic quality-compromising pre-analytical variables. So far, the only standardized phases are those from embedding on, while there is ample variation of modalities, times and protocols used in different laboratories and within the same laboratory between different specimens for all the preceding procedures. The fact that there are many variables that may cause alteration of molecular content and integrity represents a serious problem for precision medicine, where decisions about patient management are largely based on molecular data from biological specimens. Despite these issues, there is currently no requirement to control or record pre-analytical variables in clinical practice, with the single exception of breast cancer tissue handled according to the guideline jointly developed by the American Society of Clinical Oncology, ASCO, and the College of American Pathologists. It is obvious that patients should get the right answers from their medical tests. Therefore, all these variables require real-time, upfront management, because once molecular damage is done, you cannot fix it with technology. Artifactual alterations may alter data from molecular analysis, render analysis data uninterpretable, or even preclude analysis altogether if the integrity of a specimen is severely compromised. As a result, patient care and safety may be affected. In fact, molecular test results may guide clinical decisions and the appropriate use of specific therapies. In the case of companion diagnostics, molecular testing is the sine qua non for therapy allocation. The inaccuracy of a molecular test result may impact patient management in a variety of ways, and erroneous false positive or false negative test results may have significant consequences for a patient. Therefore, significant effort must be made to ensure the accuracy of test results generated in the pathology laboratory. Most of medical research is also dependent on patient leftover samples, and biomedical research itself may be compromised, including translational research and new biomarker development. There is a paradoxical gap at the moment. On one hand, testing capacity and analysis complexity is increasing, together with their efficiency, speed, and availability as the costs decrease. New technologies, such as next-generation sequencing, once limited to the research, have swiftly moved into the clinical care. There are currently more than 60,000 molecular genetic tests on the market, with 8 to 10 new products entering the market every day. Tests' quality is strictly regulated, validated, assured, and monitored. The quality of the laboratory in which the test is performed is validated and monitored. The proficiency of the professionals performing the test is rigorously enforced. On the other hand, there is considerably less focus on the fact that the accuracy of the test result still may be compromised if the quality of the biospecimen undergoing testing has been corrupted by preanalytical factors, and it is impossible building a precision medicine on a foundation of a widespread imprecision related to biospecimens. The technological capacity now exists to produce low-quality data from low-quality specimens with an unprecedented efficient technology to get the wrong answer with unprecedented speed. Everyone agrees that pre-analytics matter, that to assure reliable and biologically meaningful analysis results, the integrity of the biospecimen must be safeguarded through the clinical steps of acquisition, transport, stabilization, processing, and storage. Many studies and papers have been produced in recent years on this topic. Between 2014 and 2016, several national and international standards organizations have published guidelines for the pre-analytical steps recognized as critical for valid, reliable molecular testing, including the College of American Pathologists, the Clinical and Laboratory Standards Institute, the International and the European Committees for Technical Standardization. But compliance with any of these guidelines is voluntary, unless they are part of an accreditation program, such as International Organization for Standardization ISO 15189 Medical Laboratory Accreditation. As a result, these guidelines are inconsistently applied, either in clinical practice or research setting. For tissue specimens, the lack of any requirement to track or record the pre-analytical history of a biospecimen translates into critical pre-analytical variables being uncontrolled and undocumented for the vast majority of the specimens. As a consequence, the molecular analysis of specimens of questionable or unacceptable quality may be performed without the knowledge of the tester, producing data of questionable or unacceptable quality without the knowledge of the interpreter of that data. In 2014, the National Biomarker Development Alliance, a non-profit organization, organized a national conference to address the critical issue of uneven and unknown quality of human biospecimens in clinical medicine and translational research. The participants were more than 50 experts and thought leaders from molecular pathology, laboratory medicine, surgery, genomics, proteomics, healthcare delivery, analysis platform technology, along with payers, funders, regulators, patient advocacy, and professional societies that set and enforce standards of care, including the College of American Pathologists. The goal for the group was to come to an agreement regarding the specific variables in preanalytics that cause most of the quality compromise and molecular alteration in patient tissue and blood specimens and adversely affect DNA, RNA, and/or protein analysis results. The guiding framework for the discussion was the Pareto principle or 80-20 rule of inputs and outputs, based on which it is assumed that 80% of the problems in the outputs of a system emanate from 20% of the inputs. The goal was to identify those 20% of preanalytical variables that cause most of the variation in molecular composition and quality, and in turn most of the problems in downstream molecular analysis. The effort was focused on nucleic acids and proteins, the most commonly assayed biomolecules in both clinical and investigational biomedicine. The group agreed upon a top 6 list of key preanalytical factors for tissue biospecimens. Following this conference, the College of American Pathologists put up the Pre-analytics for Precision Medicine project team to develop a basic set of recommendations from the literature for pre-analytics for both tissue and blood specimens that could be implemented in routine pathology practice. In a 4-year effort, the team reviewed all relevant scientific data and produced a set of recommendations for these 6 key pre-analytical factors for tissue based on current literature. The key analytical factors targeted comprised cold ischemia time, method of fixation, method of processing, tissue processor variables, storage conditions, and documentation. Let's analyze in detail each one of these recommendations on key preanalytical factors. A cold ischemia time of 1 hour or less is recommended. Cold ischemia time, also referred as delay to fixation, is defined as the length of time between removal of the biospecimen from the patient and the time the biospecimen is stabilized in formalin. Let's say the biological activity in the tissue is stopped by fixing. The label cold ischemia actually refers to a room temperature environment, in contrast to warm ischemia following the vascularization of the tissue while still at a body temperature. The effects may be altered by refrigeration of the specimen. This time interval between surgical intervention and fixation of the removed specimen is crucial, since ischemia allows activation of tissue enzymes, autolysis, and degradation of proteins and nucleic acids. Data suggest that the measurable effects of cold ischemia are at least partially cancer type specific as well as biomolecule type specific and analysis platform dependent. In addition, studies comparing the effects of cold ischemia on cancer tissue and normal tissue from the same organ have demonstrated that tumor tissue is more vulnerable to the effects of cold ischemia. Substantial RNA degradation may occur during this time interval in a time-dependent manner. Cold ischemia times of several hours have minimal or no detrimental impact on biomolecular quality, extractable quantity, or molecular test results on some molecules, whereas other molecular analyses for the same specimen are altered within 60 minutes or less. Molecules may be either upregulated or downregulated, and most studies show that gene transcripts are more likely to be upregulated rather than downregulated within 2 hours of cold ischemia. Highly labile molecules, such as protein kinase and phosphoproteins, may be disproportionately affected after 30 to 45 minutes or less. Cold ischemia times of 30 minutes have been shown to increase the levels of proteins in stress response pathways, including those related to apoptosis, hypoxia, and proliferation, and may not return to baseline thereafter. Since these proteins have been implicated in cancer progression and drug resistance, their analysis may be clinically important. The effect of cold ischemia time intervals and its effect on the final assessment of prognostic markers on tumor tissue were first studied by breast pathologists. In this seminal paper dated 2009, the decrease of immunohistochemical positivity rate for estrogen and progesterone receptors by increasing the time to fixation was demonstrated. A loss of HER2 signal was evident after only 2-hour cold ischemia time with FISH, while HER2 immunohistochemistry showed no difference. By contrast, a longer time to fixation may also increase the immunohistochemical expression of a marker as demonstrated by MAP kinase on colon cancer tissue. The topic of the influence of cold ischemia on specific immunohistochemical or biomolecular determinations is still eliciting intense research. Since no single recommendation for cold ischemia time would be optimal for all tissues, all classes of molecules, or all analysis platforms, the recommendation put forward here represents a compromise that meets most current molecular testing needs for cancer patient specimens. A cold ischemia time of 1 hour is regarded as a prudent and achievable guideline and supported by the literature, and would achieve baseline standardization that would meet the requirements of the ASCO-CAP guidelines for breast cancer specimens. It is both reasonable and pragmatic to treat every specimen similarly within a uniform workflow plan rather than customize cold ischemia times for individual specimens. Additionally, actual cold ischemia times, or at a minimum deviation from the 1 hour recommendation, should be recorded in the pathology report. Methods of transfer of surgical specimens vary according to the architectural layout and distance between surgical theaters and pathology laboratories. The ideal situation is when physical location and hospital protocols allow for the immediate transfer of fresh tissue specimens for prompt grossing and/or fixation. Problems arise when the cold ischemia time cannot be kept into properly defined limits. Depending on the hospital structure and location, the transport of surgical specimens from the surgical theater to the pathology lab may prolong the ischemia time. In addition, transfer is currently performed worldwide using the most variable types of boxes, transport media, and at different temperatures. It is common practice in many hospitals to immerse surgical resection specimens and organs into large formalin-filled containers, which are then transferred to the pathology laboratory in due time, usually once a day. But formalin is highly toxic and difficult to be dealt with. The use of 10% pH neutral phosphate buffered formalin is recommended. Fixation is the first step in tissue processing. It preserves the biological tissues from degradation by inactivation of autolytic processes. A good quality fixation is very important, as it can affect the final diagnosis. A good fixation preserves the tissue components with minimal dislocation and extraction of native compounds, maintains the basic morphology and the antigenic structure, prevents biochemical alterations. In histology, buffered 10% formalin is the standard fixative. Formalin blocks degradation of tissue through stabilization of proteins with cross-linked links. It preserves a wide range of biomolecular elements as well as morphology. It is relatively inexpensive, widely available and commonly used throughout anatomic pathology practice. However, strict adherence to quality control is required to maintain the chemical quality of formalin. The pH of the formalin should be checked before used and routinely thereafter, since formalin is inherently unstable and oxidizes to formic acid. The quality of fixation depends on several variables. Fixation time is crucial. A fixation time too short or too prolonged can compromise further staining and testing. Underfixed tissues are inadequately protected against further processing reagents and present a range of artifacts. Both underfixation and overfixation of tissue compromise molecular analysis results. Nucleic acid degradation, fragmentation, and sequence alteration have all been reported as a consequence of overfixation. This damage interferes with both polymerase chain reaction amplification and next-generation sequencing. In general, the average size of DNA extracted from tissues fixed in buffered formalin decreases with increasing fixation time. There is evidence that molecular degradation in non-fatty tissue starts at 24 hours fixation time, but after about 36 hours of fixation at room temperature, excessive cross-linking and molecular damage may begin to become significant. Prolonged fixation may impair the tissue reactivity, particularly in immunohistochemical investigation. But the time of fixation is influenced by the size of the specimen, the tissue type of the specimen, the concentration and pH and temperature. It is obvious that smaller specimens fix quickly and that some tissues, like lung, being porous, favor the penetration of the aqueous formalin solution, while fat takes a lot more time to be penetrated. With such a lot of variables, it is intuitive that standardization of this process is very difficult. At room temperature, defined as 25 degrees centigrade, formalin rapidly penetrates tissue, but fixation, which means the chemical process of cross-linkage, is a slower process. Nevertheless, both tissue penetration and fixation are temperature-dependent processes. They are slowed at lower temperatures and accelerated at higher temperatures, but the rate of fixation is lower than penetration at any temperature. Tissue preparation is also profoundly affected by the pH of the formalin. The total fixation time should be no shorter than 6 hours and not more than 36 hours for most tissues. Adequate tissue fixation requires a minimum of 6 hours at room temperature. This time increases with lower temperatures. The range of 6 to 36 hours is reasonable for non-fatty tissues and may be more achievable in practice. However, a range of 6 to 24 hours may be better. Some tissues, especially those with a high fat content, such as skin or breast tissue, are an exception, and fixation times up to 48 hours may be required. Optimal fixation time for protein in most tissues is 24 hours, especially for immunohistochemical stains, whereas RNA and DNA may tolerate longer fixation times. The use of acid decalcification before or during the fixation process results in hydrolysis of DNA and RNA and is contraindicated for molecular analysis of nucleic acids. It is important to note that in histology, buffered 10% formalin is the oldest fixative used. It is still considered the best fixative, and in fact it is the standard fixative, despite the fact that it is highly toxic. When more pictograms appear on the label of a single chemical, as in this example, it's really easy to understand that it carries a serious health hazard and that exposure should be minimized. Formaldehyde, that we use in concentration of 3.7%, is acutely toxic. It can cause skin and respiratory tract irritation and corrosion, skin sensitization. It is genotoxic as well, and It has been recognized as carcinogenic type 1b in January 2016. The permissible exposure limits of the Occupational Safety and Health Administration should be observed. For example, in 2019, the European Community decided to state a maximum exposure limit of 0.3 parts per million if the exposure was lasting 8 hours a day. This limit is doubled to 0.6 parts per million for a short-term exposure. A series of protective measures should be enacted anywhere formalin is used, including the use of DPI to avoid skin and eye contact, the management of formalin only under filtered fume extractors or on aspirated special benches, monitoring regularly the airborne levels of formalin, buying pre-diluted formalin. A specific training of workers is also very important, as well as the production of written SOPs for formalin storage, spillage and waste. Another important thing that should be kept in mind is that the total fixation time should be recorded in the histology report. The thickness recommended up to 4 or 5 millimeters allows rapid and uniform penetration of formalin from the tissue surface throughout the tissue, as formalin penetration proceeds at a rate of about 1 millimeter per hour at room temperature. The ratio of fixative volume to tissue mass of at least 4:1, preferably 10:1, is recommended, with the specimen completely submerged. In contrast to the recommendation from traditional guidelines indicating a volume to mass ratio of at least 10:1, the recommendation of 4:1 represents a prudent but economical compromise that would accommodate biopsies as well as large samples to ensure that all tissue surfaces are completely enveloped by fixative. The processing of fixed tissue through dehydration to paraffin infiltration is typically automated. Multiple laboratory accreditation programs require covering the topic of tissue processor function maintenance in detail. However, strict adherence to the standardization of processing reagents is also needed to avoid specimen compromise. It is critical that reagents of high quality are used and regularly maintained. In particular, adequate maintenance of alcohols is critical. Inadequate maintenance may lead to inadequate dehydration of tissues, with consequent molecular degradation in blocks in storage due to hydrolysis. In addition, topping off of processor chambers with non-standard solutions should be strictly forbidden. Timer settings should be monitored to assure that the total time in formalin does not exceed the upper limit recommended. The use of high-quality paraffin with low melting point that liquefies at low temperatures is recommended in order to avoid the molecular damage consequent to high temperatures. In fact, using high melting point paraffins is associated with inadequate deparaffinization, reduced recovery of biomolecules from the tissue, and reduction in the extent and intensity of immunostaining. Processing involves many crucial steps where the use of different reagents over a variable period of time and at different temperatures allow fixation completion, dehydration, clearing, and impregnation. Most of the steps of processing are particularly critical for optimal tissue preservation. The College of Pathologists guidelines are intentionally addressed to 20% of the pre-analytical issues only, but there are many other relevant variables that could be easily standardized and monitored during the processing phases, including standardization of other reagents, temperature, and additional time settings. With a single machine, we are entirely running 2 of the 3 phases that we have previously defined non-standardized and at high impact. It would be easy taking advantage of the help from technology for standardizing, checking, and monitoring all the delicate steps involved in processing. Point 5. Storage conditions. Once embedded in paraffin, tissue specimens should be stored in dry, pest-free places at room temperature defined 18 to 25 degrees centigrade. Molecular degradation in stored tissue is most often due to poor fixation or inadequate processing, especially inadequate dehydration, before paraffin embedding and storage. If the tissue has been collected and processed according to a standardized, evidence-based protocol, nucleic acids and proteins extracted from formalin-fixed paraffin-embedded blocks maintained in dry, pest-free conditions at 25 degrees centigrade have been demonstrated to be comparable in quantity and quality over a time span of 1 to 12 years. Last but not least, documentation. For tissue biospecimens, documentation of data, at least of critical pre-analytical parameters of any deviations from the recommendation, should be carried out and included in the pathology report. The pre-analytical history of tissue specimens should be clearly known to judge their fitness for molecular testing. Unfortunately, documentation of cold ischemia time and total time in fixative, important determinants of molecular quality and key factors in the specimen provenance, is not routine at present. It is standard to include the fixative used in the gross description only. It is not necessary to include in the report the details of the composition and the quality of the formalin, as this should be included in a laboratory's standard operating procedures and quality control registers. This is also true for processor maintenance, paraffin type, and block storage conditions. Specimen thickness and volume to mass ratio of fixative are likely to be part of standard laboratory procedures, and their monitoring part of the laboratory accreditation program's checklists. These recommendations represent a baseline for tissue specimens from cancer patients to ensure that they are generally fit for most molecular analysis of nucleic acids and proteins. However, at the time of acquisition, it may not be known whether or not a biospecimen will be undergoing molecular testing, either as a part of immediate patient care or in the future. Therefore, it is prudent and reasonable to treat all specimens in a uniform manner that safeguards molecular integrity and ensures their fitness for molecular analysis as a routine part of patient care. It is recognized that attention to preanalytical issues has lagged behind the progression in assessment of molecular biomarkers that play a central and growing role in cancer treatment and in many other diseases. This gap is now becoming critical for precision medicine. Therefore, it is urgent that pathologists and all other health professionals who are part of the chain of custody of the integrity and quality of biospecimens implement a coordinated, evidence-based, special effort and take care to guarantee quality through all the steps of the pre-analytical phase. And with this landscape from the terrace of an old villa by the Garda Lake side, I leave the floor to Carmelo Lupo. Carmelo Lupo is Head of Innovation in Diapath, Italian company that designs and develops instruments and reagents for pathology labs worldwide. With 20 years of experience in pathology and molecular pathology labs as a laboratory manager and contract professor of a Master in Laboratory Medicine, Carmelo is dedicated to overviewing the product concept design, product development and prototype testing, and develops professional relationships with worldwide health professionals to understand the problems and collaborate to find solutions. For these reasons, he leads scientific initiatives and manages international projects and collaborations with research institutions and universities.
Thank you, Antonella, for your interesting presentation. As you clearly explained before, we are now in the precision medicine era, where the value of information has a high impact on accurate diagnosis and on definition of correct and personalized treatment for patients. The main goal is to achieve the appropriate care for every patient. In this field, the role of anatomic pathology is of the utmost importance for tissue management in the formulation of diagnosis. Here, I would like to share an overview on the state of art on evolution of the technology in histopathology and molecular pathology. In histopathology, we are now focusing on tissue processing. That is a multi-step process where a transformation of biological specimen occurs from water-based phase to paraffin-embedded. It is an irreversible, essential process where information related to potential mutation or to protein or gene expression do not have to be damaged in the process. Regarding molecular pathology, we are not talking about any transformation, but about the detection of information. This detection phase depends totally on the quality of the previous tissue processing, therefore on the quality of the transformation step. What about technology? What about risk management? The evolution of molecular pathology technology is bringing an important increase in the number of information that can be obtained and to the parallel reduction of costs, resources and time needed. It is important to remember also that the technology is going in the direction of standardized process, no more relying on the single user expertise. To avoid variability and potential human mistakes, the manual steps and human intervention for molecular analysis are more and more reduced, together with agreed control in terms of batch, process, and reagent control. Referring to the previous slide, Antonella presented you the high-impact steps in pathology. I would focus on the red square of tissue processing. From the 18th century up to present, there was a great increase in the knowledge and literature production. In all this time, tissue processing has always been as a sequence of steps of chemicals, namely formalin, alcohols, clarifying agents, and paraffin, with no great changes, but the fact the process moved from manual to semi-automated and to automated. So, how could we define tissue processing today? Exactly in the same way as you could define it many years ago. In this slide, we can see the literature production represented by about 4 million publications, with a significant increase in the last years. It is clear the technology gap between the two diagnostics areas. How can be sure that an automated process is a perfect process? Is it always standardized in the same way everywhere? In this kind of automated process, there is still the chance of machine failures as well as human mistakes. Can the actual technology guarantee the appropriate reliability for daily process for every single patient sample? I will provide just 2 examples for the major problem that can occur. Which instrument works alone without any professional in overnight process in an empty histopathology lab with few hundreds of unique and irreplaceable samples inside? If you think about it, all the other instruments of lab like microtomes, woods, stainers or microscopes are active when the lab staff is present. If a problem occurs, you can immediately visualize it and try to solve it, or in case, you can call technical assistance for help. None of the instruments above transform specimen in an irreversible way. In the best case scenario, you will have a diagnostic delay. In the worst case scenario, specimen will be lost. Paradoxically, in the instruments that has the highest impact on samples can only send a message in case of problems. It is obvious, but that in the middle of the night, it's very difficult to provide an efficient corrective action. Reagent mismatch. It is true that on the labels there are the names and pictograms to recognize properly each reagent that is contained, but it can happen that during the change of the tissue processor, the operator can make mistakes. All reagents are transparent and look similar, but their function during the processing is specific. It can happen that they are loaded on the machine in the wrong order, and in this case, all the process is irreversibly compromised. How can we avoid those problems? How can technology help us? Regarding the first example about the possibility of overnight processing failure, the technology allows the use of an algorithm-based system of artificial intelligence. This allows to manage promptly failure problems in order to avoid that samples are irreversibly damaged. In the second example, the RFID technology can transfer in an unambiguous way many information, such as the type of chemical, batch number, expiring date, to the machine that can in this way check if the reagents are correctly replaced. In the 21st century, most of the tissue processors on the market are not equipped with any artificial intelligence-based system. In case of failure, they simply stop, not being able to manage the failure occurred. As a common washing machine, can we really trust a washing machine for unique and unrepeatable samples? Intelligence has been defined in many ways: the capacity for logic, understanding, self-awareness, learning, emotional knowledge, reasoning, planning, creativity, critical thinking, and problem solving. More generally, it can be described as an ability to perceive or to infer information. Intelligence is often studied in humans, but it has also been observed in both animals and plants, despite controversial opinions if these forms of life exhibit intelligence. Intelligence in computers or in other machines is called artificial intelligence. How could an algorithm help us? Because an algorithm is a succession of instructions or steps that defines the operations to be performed on the data in order to obtain the results. An algorithm provides the solution to a class of problems. The last keyword is evolution. Evolution is a process based on mutations. The mutation is the key to our evolution. It has enabled us to evolve from a single-celled organism into the dominant species on the planet. Its process is slow and normally takes thousands and thousands of years. One driver for automotive evolution is the safety of passengers. In the same way, the evolution of tissue processor should follow the main aim of sample safety. Another example is from aerospace industry. The processing is a basic and crucial step. During this process, you should check a number of conditions that will determine irreversibly the right fixation and processing of surgical specimens. The correct processing of surgical specimens will be critical for the morphological, immunohistochemical, and molecular analysis. The quality of surgical specimen can heavily influence a diagnosis. Wrong or improper fixation and processing can alter the morphology, create false positives and or negatives in methods such as AHC, ISH, and influence the molecular analysis. This strongly impacts on diagnosis and indirectly on the life of the patients. It would be nice if any of you would like to share their successful experiences where you were able to fix an issue and to guarantee the quality in the pre-analytical and analytical phase. For this reason, we would like to organize in the near future a webinar in which you will have your own space to share your stories. All of you that participate to this webinar will receive an email from Diapath with an invitation to candidate as a speaker.
I'm very glad to invite you to the next webinar, Quality Control in Immunohistochemistry, which is due on the 22nd of April 2021 at 10 a.m. or 4 p.m. CET. You're welcome. Subscribe under www.diapath.com/webinars-list.